Hi, this is Dr. Lonaville. We're going to isolate some genomic DNA. So genomic DNA isolation is really similar to how we're going to isolate RNA later, but RNA has got some extra sorts of things you have to worry about in terms of being clean and avoiding contamination, and also avoiding RNases. DNA is super stable. And so you have to worry about that less, but you do want to wear gloves just to avoid contaminating your genomic DNA with human DNA. So human DNA from like facial hair or skin cells or anything like that might fall into your sample. Gloves can minimize that. So I'm going to start with my worms that I collected off a plate. Concentrated them by allowing them to settle and pellet, and then sonicating them. You saw that on another video. And then the first step in this kit, we're just going to use a kit to isolate genomic DNA, is I'm going to add 400 microliters of the genomic lysis buffer to the sample. So the genomic lysis buffer has got beta-mercaptan in it, and that is very, very stinky. Beta-mercaptan will denature proteins and break disulfide bonds, and you're particularly sensitive. Your nose is particularly sensitive to it, so we're going to do that part in a hood. Here we are in the hood, and I got my sample here and my genomic lysis buffer, which is stinky. That's why we're in the hood. And it's also a little bit foamy because it's got some detergent in it that is going to help pick up the cells. And I'm just going to add that 400 microliters to my tube right here. So the idea is you want to open up those cells, and you want to denature the proteins that are wrapping up the DNA in those cells. So it should be pretty clear sort of fluid, where it was sort of cloudy before. Okay. And now I'm going to do is I'm going to add that to the column. Here's the column. You might be looking for something bigger than that if you were in like a chem lab or something like that. But this little white pellet there looks like an aspirin tablet. That's actually what the DNA is going to bind to. It's made from the skeleton of diatoms. It's called diatomaceous earth. It's essentially glass. And glass has a positive charge on it. The phosphates on the DNA has a negative charge on it, so this will collect the DNA. It's really important whenever you're handling these columns that you don't touch that little disc down there with your pipette tip, because then things can go through it without binding to it. So you want to deliver to right on top of that column when you're delivering, but not actually touch it. And then to assemble it, you just put it in a tube. These are microfuge tubes just without the cap. So assembling the column, I'm just going to put this on top of this, and then I'm going to put my sample on the column. Okay, again, I'm paying attention to not touching that little white disc at the bottom of the column, but I don't want to just hit the sides. I want to deliver it right to the middle of the tube. Got some bubbles there. Want to get the whole sample on there, because that'll mean I'll get the most DNA I can get. Okay, and then this will go chem waste, and then this right here, this assembly is going to go onto the centrifuge. Okay, taking a look at the centrifuge, there's various centrifuges in the lab. This one is pretty typical, has a little button right here to open it up, and power's got to be on to open it up. So the important thing is you want to balance this centrifuge. So you can see here, there's my sample with an X on it, and then this is just an empty column. That's going to be very similar in weight to this one with my sample in it. I could do even better by putting 400 microliters in this column, but I don't want to waste this column. So it's important that these are balanced against each other. This is going to generate about 5,000 times the force of gravity when it spins. We'll talk more about that in another video. But if you are off by a lot, say 1/10 of a gram or something like that, then it's pulling harder on one side of the centrifuge than another side of the centrifuge as it spins around, and that can damage the centrifuge. In an ultra centrifuge, which we're not going to use in this class, you get up to 100,000 times the force of gravity, and so a very small difference in the weight of the tubes on opposite sides of the centrifuge can turn into a really big force. So for right now, I'm just going to cover these, and I'm going to centrifuge this for a minute at full speed, just the maximum RPMs that's this centrifuge will handle. Okay, now this is centrifuged, and you can see that the liquid is down here in the bottom of the tube. The DNA is stuck to that white column, okay, right, that little thing that looks like an aspirin right there. And now what we're going to do is we're going to wash that DNA to get rid of things we don't want, like carbohydrates, proteins, fat, etc. So we're just going to put this back on here, and now I'm going to add 200 microliters of the first wash, the pre-wash buffer. I'll show it right here. DNA pre-wash buffer, and centrifuge that for 1 minute at full speed. Okay, this is after the first wash. You can see I have more fluid in there. The next wash is 500 microliters of the GDNA wash buffer. And if I add 500 microliters to this, it'll cover the bottom of the tube. So I'm going to empty this bottom tube into the liquid waste that'll be living in the hood. Okay, just tip that into there, and then you use the same tube. Put that back on here, and then add your 500 microliters of the GDNA wash. Okay, again, I'm going to be careful not to touch the white column on the bottom of this thing. Put that 500 microliters in, and it goes back into the centrifuge for another minute at full speed. Okay, I've washed with the GDNA wash, and I've got my 500 microliters in the bottom of this tube. I'm going to add this. I'm going to empty this into the hood liquid waste, and then it's important that I put this back together empty, so there's nothing on the top and nothing on the bottom, and I still spin it again for another full minute at high speed. And what this is doing is getting rid of any residual liquid that was on that white column. And that's important because there can be ethanol left over from these washes that is on the column. You don't want ethanol in your DNA because it can inhibit PCR, which is the next thing we're going to do. And so this step is really important, that you spin it for a minute empty. Okay, now I've got my DNA should be dry and clean. I'm going to remove this bottom collection tube, and I'm going to put this column onto a new clean microfuge tube that will contain my DNA when I'm all done. So at this point, I am ready to elute the DNA off that little column. You can use molecular grade water. Tap water, distilled water can have enough contaminants in it still that it can inhibit PCR. So we actually buy water that is molecular grade, super clean water that's not going to be inhibiting PCR. So you can use water to elute your DNA, or you can use what we're going to use today, which is the DNA elution buffer that comes in the kit. The DNA elution buffer contains EDTA, which is a calcium chelator, and most enzymes that chew up DNA, DNases, require calcium for digesting DNA. So by removing calcium from the environment, you can prevent your DNA from degrading. The reason you would use water instead of this is EDTA can inhibit some downstream reactions, some things that you would want to do with your DNA. So if you know that ahead of time, you would use water instead of EDTA. So you can, the kit says, this particular kit says you can use up to 50 microliters to elute your DNA. I'm having you use 40 microliters. It's always a balance. So 50 microliters is going to flush the most DNA out of your column, but then your DNA will be less concentrated when it comes off the column. 40 microliters might get a little less DNA off the column, but it'll be more concentrated, and that's the trade-off we're going to make today. Okay? So I'm going to put my 40 microliters on that column, directly on top of the column, but again, not touching the column. And then I'm going to let it sit for 5 minutes to make sure all of that fluid gets to the DNA, and then the DNA is ready to be knocked off that column. Okay, so I'm gonna drop it right on top without touching, like that, and then I'm just gonna let it sit for 5 minutes, and then I'll spin it for a minute. Okay, that is our DNA right there. I still have my column on top of it. Now we are going to measure the concentration of that DNA on the nanodrop, which is in the molecular commons, and your TA will take you up there. One thing I forgot to mention is when you are doing this last spin, you probably want to remove the top of the microfuge tube. Just pull it off like that, because otherwise that top can break off while it's spinning. Okay, so now let's see. I'm gonna show you the nanodrop. Okay, here is the nanodrop over in the molecular commons. I'm gonna pan over here. So there it is right here. Okay, this is the machine. And what we're going to do is we're going to calibrate this for the elution buffer. So whatever you use to elute your DNA, you got to tell the machine, you got to blank it with that thing. So if it's molecular water or this DNA elution buffer, you got to blank the machine. So what the nanodrop is just a spectrophotometer that doesn't use a cuvette. Instead, it uses a little drop of liquid right here, and I'll zoom in on that so you can see it. And it'll form a column with the top of this column, and that acts like the cuvette. So, and just measures absorbance to that little bead of water. So I'm gonna put 1 microliter of our sample DNA elution buffer on there, and this is a big advantage of the nanodrop because you don't waste a lot of your sample. So you'll see me adding the 1 microliter. There's a little black dot. I'm going to add it right to there. You can touch it. Okay, just 1 microliter. Gently put this down, and then okay, I'm going to say blank. So I'm going to call that blank. You hear that little click? That's how it's forming the column that it's going to look at. Okay, now it says this is a blank. And now what I can do is I can put my sample on there and find out what the concentration of the sample is for DNA. So I'm doing the same thing. I'm just gonna wipe off the surface of that 1 microliter, and now I'm gonna put 1 microliter of my DNA sample on there. Again, gently close it. And now I'm gonna say measure up here. Okay, so we got some DNA. So the concentration is 32.6 nanograms per microliter. That's not a lot. It's not too little. It's okay. DNA and RNA will have a maximum absorbance at 260 nanometers, and protein will have a maximum absorbance at 280 nanometers. If you look at the ratio of 260 to 280, it should be 1.8 or above. Okay, this number here. Okay, and it's 1.93, so that's good. The 260 to 230 ratio should be around 2, and it says 0.09, which is not great. So there is leftover ethanol in my sample, and as a result of that, I might have some trouble doing PCR. I'm only going to use about 3 microliters of this sample in my PCR reaction out of 20. So any ethanol that's in there shouldn't massively inhibit the PCR reaction. We'll just see if it works. Okay, so you would want to record all these numbers for your DNA sample, and you would also want to take a picture of your spectrum that you got here with your DNA sample. So now I have that concentration of DNA in nanograms per microliter. I'm going to use that in figuring out how much I put into a PCR reaction. It's really important for PCR that you use these small PCR tubes that are super flexible and small. Compare that to the size of a regular microfuge tube. Okay, we're going to give you these tubes. So you're going to set up your PCR reactions in these small tubes. If you set them up in the big tube, your PCR reaction won't work. So the whole idea is that the heat of the machine transfers to your sample really efficiently. These are too big and too thick to do that. So you want to make sure that you use these. Also on these samples, you cannot write on the dome, okay, on the lid, because there's a heated lid that's going to come down on top of that, and it'll just burn off whatever marking. So you can write on the little lip, you can write on underneath the edge here, this top edge, but you can't write here on the conical part, and you can't write on the dome. That'll just disappear. So that's actually super important, because lots of people in this lab, you're going to have to be able to see which is your sample and which is their sample. And also within your samples, you're going to need to know what is the positive and what's the negative control. Even if you find your tubes, but you can't tell what's positive and what's negative, you really can't interpret your results. Okay? So that should get you started. Pretty simple lab.